Somewhere in the Pacific Ocean, about a mile below the surface, a fish is hunting. It is carrying a light. The light is part of its body. It grows directly from its head, and it is glowing in water that has been in complete, total darkness since before humans existed. Down here, that is not even close to the strangest thing. Below 1,000 meters, sunlight is completely gone. The pressure is enough to crush a submarine. The temperature sits just above freezing, and food is so scarce that some creatures go weeks, sometimes months between meals. This environment is not an edge case. It is the largest living space on Earth, and most of it has never been seen by human eyes. The animals living in it were not always like this. Their ancestors were ordinary fish, surface fish, fish that followed the same rules as everything else. Then over millions of years, the deep sea got hold of them, and it remade them into something else entirely. There are fish down here with no eyes at all, not damaged, not small, gone, because in permanent darkness, eyes are a cost with no return. There are fish with eyes mounted inside transparent domes on their heads, rotating, scanning, locked onto movement happening above them in the dark. And there are predators that do not chase, do not search, do not move. They simply hang in the darkness, perfectly still, dangling a light, waiting for something to come to them. Every one of these changes has a reason. Every strange feature is a response to the same relentless pressure, and the deeper you go, the further that pressure pushes life away from anything recognizable. Tonight, we look at how the deep sea reshapes ordinary fish into forms that follow a completely different set of biological rules, and why those changes go as far as they do. There is a way to divide the ocean that has nothing to do with maps or continents, a structure defined instead by light, pressure, and the slow shifting of energy as depth increases. It is not a boundary you can see from above, but it shapes every living thing beneath the surface with quiet precision, layering the water into distinct realms, each with its own rules for survival. Closest to the surface lies the sunlight zone, extending from 0 to 200 meters. Here, light is abundant and stable, and nearly all familiar ocean life exists within it. Plants drift and grow, microscopic phytoplankton convert sunlight into energy, and food chains build upward in complexity. Motion is constant, color is visible, and life is driven by the steady input of solar energy. This is the ocean most closely aligned with the rhythms of the land above. Below it, the twilight zone stretches from 200 to 1,000 meters, a region where light begins to thin and weaken. Blue wavelengths linger the longest, fading gradually until even they dissolve into darkness. Shapes lose definition, colors flatten, and visibility becomes unreliable. It is here that the first structural changes in life begin to appear, subtle at first, then increasingly pronounced as light continues to retreat. Beyond 1,000 meters, the midnight zone begins. No sunlight reaches this depth. It has not reached here for the entire history of complex life on Earth. Temperatures hover just above freezing, and pressure rises beyond 400 atmospheres, compressing every surface and altering the behavior of molecules themselves. Vision loses its relevance, and energy becomes scarce. Life persists, but it does so under entirely different constraints, operating slowly, efficiently, and with minimal waste. Deeper still lies the abyssal zone, between 4,000 and 6,000 meters. This is a vast, quiet expanse that covers more than half of the planet's surface. The seafloor here is broad and continuous, broken only by occasional ridges and rises. Movement is rare, and encounters between organisms are infrequent. The environment is stable over long timescales, with little variation in temperature or chemistry, creating a world where change happens almost imperceptibly. At the greatest depths from 6,000 to 11,000 meters, the hadal zone descends into oceanic trenches. These are narrow, steep-walled depressions where pressure reaches its most extreme levels, and the physical limits of biology are tested. Conditions here are not just intensified versions of what lies above. They represent a fundamental shift where survival requires solutions that do not exist anywhere else in the ocean. The average depth of the ocean is 3,688 meters, which means that most of the ocean already exists in complete permanent darkness. The surface, bright and active, is only a thin layer stretched across an immense volume of cold, pressurized water. By sheer size, the deep sea forms the largest habitat on Earth, larger than all forests, grasslands, and shallow marine environments combined. Yet the vast majority of it remains unseen, with 95% never directly observed by human eyes. Across all these layers, three forces remain constant, shaping every organism that lives within them. Pressure increases with depth, altering the structure of cells and the chemistry of life. Cold slows metabolism and extends biological processes over longer timescales. Darkness removes the influence of sunlight entirely, forcing life to rely on alternative sources of energy and entirely different ways of sensing the environment. Every adaptation that appears in the deep sea is a response to these forces acting together, not in isolation. The structure of the ocean is not simply a descent into deeper water, it is a gradual shift in the rules that govern life, beginning not at the bottom, but at the moment the light begins to fade. At a certain depth, brightness stops behaving like a constant and begins to thin into something fragile, a fading presence that no longer fills the water but lingers in it. The descent into the twilight zone begins around 200 meters, where the surface above is still visible in principle, but no longer reliable. Light arrives weakened, scattered, filtered into narrow bands that lose strength with every meter downward, until what remains is no longer illumination in the usual sense, but a dim residue, a trace of something that once defined the entire ocean. By 600 meters, this residue has narrowed to a faint blue glow, suspended above like a distant ceiling that continues to recede. Objects lose their edges, then their color, then their distinction from the surrounding water. The boundary between organism and environment begins to soften, and the simple act of being seen becomes a problem that shapes bodies from the inside out. By 1,000 meters, even this last fragment disappears entirely. But before that final absence arrives, a transformation takes hold in the dim interval where light still exists but no longer behaves as expected. Within this fading band, a particular kind of illusion emerges, not as behavior, but as anatomy. The hatchetfish carries it in its form, a body compressed from side to side into a thin, blade-like shape that presents almost no surface when viewed from below. Its underside is lined with photophores, small organs that produce light calibrated with remarkable precision. They emit a glow that mirrors the intensity and color of the light filtering down from above, matching it so closely that the fish becomes indistinguishable from the faint brightness overhead. From below, where predators search upward for silhouettes, there is nothing to resolve. The expected contrast between body and background is erased. The fish does not hide behind an object or within a structure. It dissolves into the field of light itself, replacing its shadow with an imitation of the sky. This is not an action performed in response to danger, but a condition of existence, a body built to remove its own outline from perception. This solution appears again and again throughout the twilight zone, arising independently in species that share no close ancestry. The problem remains constant across this depth, the faint glow above revealing anything that interrupts it. The answer converges toward the same principle, producing light where darkness would otherwise give shape away. Evolution arrives repeatedly at this arrangement, refining the balance between emission and environment until the boundary between organism and water becomes almost impossible to detect. Elsewhere in the same dim water, a different form takes shape, one that does not erase its presence, but prepares to use the remaining light in brief, decisive encounters. The viperfish drifts with a posture that appears restrained, its long fangs curving upward along the outside of its head because they cannot be contained within its closed jaws. These teeth extend past the limits of what its skull can accommodate, forming a structure that is less about enclosure and more about interception. Behind the skull, the first vertebra is reinforced in a way that alters the movement of the entire head. When the fish strikes, the force of impact is absorbed along this modified joint, allowing a rapid forward motion that drives the teeth into prey with minimal resistance. The mechanics are not exaggerated but precise, distributing stress through a structure built to handle sudden acceleration in dense water. A slender extension projects forward from its body, bearing a small photophore that emits a point of light just ahead of the mouth. This light does not fill the surroundings, but occupies a narrow space, enough to draw attention within the limited visibility that remains. It creates a focus point, a small disturbance in the dim field above, inviting movement toward it. In this zone, where light is nearly gone but not entirely absent, these strategies unfold within a narrow margin between visibility and concealment. The water still carries a faint trace of illumination, and within that trace, bodies either remove themselves from detection or position themselves to intercept what drifts too close. As the last of the light fades completely, the conditions that shaped these forms give way to something else, and the structures that follow no longer rely on imitation or alignment with any external source. Pressure is not felt all at once in the ocean. It accumulates quietly, layer by layer, until it becomes the defining condition of everything below. At the surface, the body exists under a single atmosphere, the familiar weight of air pressing evenly in all directions. It is a pressure so constant it disappears from awareness, shaping physiology without drawing attention to itself. As depth increases, that hidden force multiplies with steady precision, building into something far less forgiving. At 1,000 meters, the surrounding water exerts 100 atmospheres, compressing every surface, every cavity, every microscopic structure. The increase is not abstract but physical, a direct multiplication of force applied to every part of the body at once. Far below, at the deepest known points of the Mariana Trench, pressure reaches 1,086 atmospheres, a load comparable to 50 jumbo jets balanced on a single square meter. It is a magnitude that would collapse most engineered structures, yet within it, living tissue continues to function. What this pressure does is not immediate destruction, but gradual interference with the smallest systems that sustain life. Cell membranes, composed of delicate lipid layers, are compressed, altering their fluidity and permeability. Proteins, which rely on precise three-dimensional shapes to carry out chemical reactions, begin to fold incorrectly or lose stability under compression. Enzymes, responsible for catalyzing the reactions that power movement and metabolism, become less effective as their structures distort. The entire biochemical network slows or fails if left uncorrected. The deep sea does not crush its inhabitants because those inhabitants have changed at the molecular level to meet this force. Their bodies are not resisting pressure in the way a rigid object resists weight. Instead, They allow pressure to pass through them while maintaining internal stability through chemistry. Among the most important of these adaptations is a compound known as trimethylamine oxide, or TMAO, a small molecule that accumulates in increasing concentrations as depth increases. TMAO interacts directly with proteins, stabilizing their folded structures against the compressive forces of the surrounding water. It counterbalances the distortion that pressure would otherwise impose, preserving the shapes required for biological function. This adjustment is not superficial, but embedded within the chemistry of the organism itself. The deeper a fish lives, the higher its internal concentration of TMAO, forming a gradient that mirrors the depth of its habitat. From this, scientists can infer a fish's typical range by analyzing a single tissue sample. The concentration of TMAO serves as a record written into the organism's cells, a chemical indicator of the pressure conditions it has adapted to over generations. Yet this solution carries a limit. Beyond a certain concentration, TMAO begins to interfere with other cellular processes, disrupting the balance it was meant to protect. This creates an upper boundary, a point beyond which further adaptation becomes chemically unstable. That boundary appears near 8,336 meters, the depth at which the deepest known vertebrate has been observed. The snailfish Pseudoliparis swirae inhabits this region, moving through water where pressure approaches the maximum tolerable limits for complex life. Its body reflects the constraints imposed by this environment. Its bones are only lightly mineralized, retaining flexibility that prevents fracture under compression. Its tissues are soft, almost gelatinous, allowing external pressure to equalize without resistance. Within its cells, the composition of membranes shifts toward a higher proportion of unsaturated fats, maintaining fluidity where typical lipids would stiffen and lose function. These adjustments ensure that essential processes continue without interruption, even as external pressure reshapes the physical context of every molecule involved. The result is an organism that appears fragile, nearly translucent, with a structure that seems incompatible with endurance, yet it persists precisely because of that softness. In this way, pressure does not sculpt life into strength, as it might in other environments. It favors flexibility, chemical balance, and structural minimalism, producing forms that appear reduced, but are in fact precisely tuned to survive within extreme compression. Light returns here in a different form, not as a distant glow filtering down from above, but as something generated within living bodies, appearing in brief pulses and steady glimmers that have no connection to the surface. It emerges from tissue itself, a phenomenon that has arisen again and again across unrelated lineages, each arriving at its own method, yet converging on the same quiet result. In the absence of sunlight, life has found a way to produce its own illumination. This capacity has appeared at least 40 separate times in marine organisms, making it one of the most repeated innovations in the history of evolution. It is not a single pathway reused across species, but a collection of distinct chemical systems, each built from different components, each refined independently. Despite this diversity, they all lead to the same outcome, the creation of light without heat, a cold radiance that carries energy with remarkable efficiency. At the center of many of these systems lies a reaction involving a molecule called luciferin and an enzyme known as luciferase. When luciferin encounters oxygen under the influence of luciferase, energy is released in the form of visible light rather than thermal waste. The reaction proceeds with minimal loss, converting nearly all available energy into illumination. Compared to a standard light bulb, which transforms only a small fraction of its energy into light while dissipating the rest as heat, This process operates with a precision that seems almost complete. The color of this light is not arbitrary. In seawater, shorter wavelengths travel further before being absorbed, and so most deep-sea organisms produce light in the blue-green range, a spectrum that carries efficiently through the surrounding medium. These wavelengths extend the reach of a signal, or lure, allowing it to move outward into darkness with minimal attenuation. The ocean shapes not only the existence of light, but also its specific form. Yet within this pattern, there are exceptions that depart from the shared solution. The dragonfish, Malacosteus niger, occupies depths where even the faint traces of ambient illumination no longer exist, and within its body, a different kind of light is produced. It emits a deep red glow, a wavelength that is quickly absorbed by seawater and remains invisible to most organisms at that depth. At the same time, it possesses specialized photoreceptors capable of detecting this otherwise hidden color. The result is a channel of perception that exists only within the organism itself. It can illuminate its surroundings in a spectrum that others cannot perceive, observing movement and form without revealing its presence. The water around it remains unchanged to any observer lacking the necessary sensitivity, while within its own field of vision, shapes are outlined in a private hue. This arrangement does not alter the environment, but it alters the relationship between the organism and that environment in a precise and isolated way. Not all deep-sea fish generate light through internal chemistry. Many rely on symbiotic relationships with bioluminescent bacteria housed within specialized organs that provide nutrients and protection in exchange for a steady emission of light. These bacterial communities live within the body of the host, their metabolic processes producing illumination that the fish cannot create on its own. The partnership is stable and continuous, a shared system in which neither participant functions independently. In some species, this borrowed light becomes inseparable from the organism's identity. The anglerfish carries a luminous structure that depends entirely on its resident bacteria, a glow that persists without interruption because it cannot be regulated in the way internally generated light can. The illumination is constant, a presence that exists as long as the relationship is maintained, embedded within the biology of both host and symbiont. Across these variations, light becomes more than a simple adaptation. It is integrated into communication, detection, and interaction, woven into the structure of the body and the chemistry of cells. It replaces what was lost when sunlight disappeared, not by replicating it, but by redefining how illumination can exist within a living system. It begins as an imbalance, a difference so extreme that it reshapes the entire life cycle of a species. The female anglerfish moves through the water with a body built for capture, her length reaching 20 to 30 centimeters, her mouth wide enough to enclose prey larger than herself, her teeth long and narrow, angled inward so that anything taken in cannot escape. The luminous appendage that extends from her head glows steadily, a small, constant presence in the surrounding dark. For a long time, this was the only form ever encountered. Specimens collected over decades showed no variation that suggested a counterpart. The assumption settled quietly into place that males were either rare beyond expectation or simply absent from the records for reasons no one could explain. The species appeared to exist in a single visible form, complete in itself, with no clear indication of how reproduction occurred within such an arrangement. Closer examination of preserved individuals revealed something that did not fit this understanding. Attached to the bodies of some females were small growths, irregular in shape, embedded along the surface of the skin. They were first described as parasites, simplified organisms that had lost most of their original structure and now depended entirely on a host. The appearance supported this interpretation, reduced bodies with no clear resemblance to the larger fish they accompanied. The revision came slowly, through anatomical study that traced connections deeper than the surface. These structures were not foreign organisms in the usual sense. They shared the same lineage as the host, and their reduced form was not degeneration but transformation. They were the males, present all along but altered to a degree that concealed their identity. A male anglerfish begins life as an independent organism, but only briefly. At birth, he is extremely small, often less than a centimeter in length, with a body that contains only the systems necessary to locate a female. His digestive tract is incomplete, unable to support long-term survival. He exists in a narrow window of time, sustained by the energy reserves he carries, moving through the water guided by chemical cues that disperse from a distant source. The environment offers no landmarks, no gradients of light to follow. Instead, he relies on pheromones, faint molecular traces that drift through the water column. These signals spread unevenly, forming trails that can extend across considerable distances. The male follows them with a sensitivity tuned to detect minimal concentrations, adjusting his path in response to gradients too subtle to measure directly. When he reaches the source, the process shifts from navigation to contact. He attaches to the female by biting into her skin, initiating a reaction that alters both bodies at the point of connection. Enzymes break down the outer layers of tissue, allowing the two organisms to fuse at a cellular level. Blood vessels merge, and the male becomes integrated into the female's circulatory system, receiving nutrients directly from her body. From that moment, his structure begins to change. Organs that are no longer required are gradually reabsorbed or reduced. The eyes diminish, the fins recede. The internal systems that once supported independent movement and function disappear. The transformation does not occur abruptly, but unfolds over time, as the male transitions from a separate organism into a permanent extension of the female. What remains is a reproductive unit, a pair of testes embedded within her body, connected to her bloodstream and capable of providing genetic material when needed. The rest of the original structure is gone, not as a loss imposed from outside, but as a consequence of a life cycle that prioritizes certainty over independence. In some cases, multiple males attach to a single female, each occupying a small region of her surface, all sharing the same integration into her physiology. This arrangement does not fit easily into familiar categories. It is neither a temporary association nor a typical form of parasitism. The male does not simply draw resources from the female while maintaining his own identity. Instead, his identity dissolves into the shared system, leaving behind only the function that remains necessary. The transformation reflects the conditions of a habitat where encounters are rare and unpredictable, where the probability of meeting more than once may not exist within a lifetime. In such a setting, permanence replaces repetition. The moment of contact becomes the only opportunity, and the biology that follows ensures that it is never lost. Vision lingers here in forms that do not follow the expectations set by the surface, where light defines what can be seen and how eyes are built to receive it. In these depths, where illumination is intermittent or nearly absent, the logic of sight bends into unfamiliar shapes, preserving its function in ways that seem to contradict the conditions surrounding it. Instead of fading away, it becomes more specialized, more precise, adapting not to abundance but to scarcity. The barrel-eye fish, Macropinna microstoma, carries one of the most unusual structures ever observed in a vertebrate. The upper portion of its head is enclosed by a transparent dome not a rigid skull, but a fluid-filled shield that allows light to pass inward with minimal distortion. Beneath this clear surface sit two elongated, tubular eyes, positioned side by side and oriented upward. They are not fixed in place. Each eye can rotate within the dome, shifting its angle to adjust the direction of focus. At first glance, the front of the fish appears to hold a pair of conventional eyes, dark spots set into the face. These are not visual organs at all, but nostrils, positioned where eyes might be expected. The true eyes remain hidden within the dome, their orientation determined by the demands of the environment rather than by symmetry or familiarity. When the fish drifts beneath potential prey, the eyes angle upward, tracking faint outlines against what little brightness filters down from above. When prey moves closer, the eyes rotate forward, aligning directly with the target. For many years, this capability remained unknown. Specimens brought to the surface lost the integrity of the dome, which collapsed under reduced pressure, obscuring the internal arrangement. It was only in 2009, when a living individual was recorded in its natural environment, that the full function of this system became visible. The movement of the eyes within the transparent enclosure revealed a design that could not be inferred from preserved samples. Elsewhere, a different solution appears in the spookfish, Dolichopteryx longips, whose visual system departs entirely from the standard lens-based structure found in other vertebrates. Instead of relying on curved lenses to focus incoming light, it uses reflective surfaces composed of microscopic crystalline plates. These plates are arranged in a mosaic that redirects light onto the retina, functioning as a mirror rather than a lens. This approach, common in some invertebrates, is otherwise absent from vertebrate evolution, yet here it emerges fully formed, solving the problem of light collection through an entirely separate pathway. The diversity of these systems extends further in the deep-sea squid, Histiotuthis heteropsis. Its two eyes differ significantly in size and function, each adapted to a different direction and type of signal. One eye is larger, with a yellow pigmentation that enhances sensitivity to faint flashes originating below. The other is smaller, tuned to detect shapes against the dim background above. Together, they divide the visual field into two distinct zones, each processed by an organ specialized for its specific task, allowing the animal to interpret multiple layers of information simultaneously. In some species, light detection extends beyond the eyes themselves. The dragonfish possesses photophores along its lateral line that may act as additional sensors, capturing reflected light through the skin. These structures blur the boundary between vision and touch, integrating light perception into the broader sensory system of the body. Instead of relying solely on forward-facing organs, the fish gathers information from multiple directions, creating a distributed awareness of its surroundings. Across these variations, vision becomes less about forming a clear image and more about detecting the smallest possible signal within an environment that offers very little to work with. Each structure refines a different aspect of that task, bending the rules of anatomy to preserve a sense that might otherwise be lost. Sound, movement, and chemistry take on a greater presence, where sight offers little guidance, forming a layered field of information that surrounds every organism. In this environment, perception does not rely on images, but on patterns carried through water itself, subtle disturbances that spread outward and linger long enough to be detected by systems tuned to their smallest variations. Along the length of a fish's body runs a structure known as the lateral line, a continuous network of sensory cells embedded just beneath the skin. These cells, called neuromasts, respond to changes in pressure and motion within the surrounding water. As currents shift or an object passes nearby, the resulting displacement creates waves that move through the fluid, bending the tiny hair-like structures within each sensor. The pattern of this bending encodes direction, distance, and speed, forming a spatial awareness that does not depend on light. Through this system, a fish perceives the water as a textured medium rather than an empty space. Obstacles alter the flow around them, leaving detectable signatures that can be followed or avoided. Other organisms generate their own disturbances as they move, each motion producing a trace that spreads outward and can be read by any nearby observer equipped to interpret it. Even in complete darkness, a fish can navigate with precision, responding to these subtle cues as reliably as if it were using sight. Experiments conducted in controlled conditions have shown that fish deprived of vision retain the ability to move through complex environments without collision. With their eyes covered, they continue to detect barriers, track motion, and adjust their path in response to changes in water flow. The lateral line functions not as a supplement to vision, but as an independent sense capable of guiding behavior in the absence of any visual input. Another layer of perception emerges through the detection of electrical fields. Certain deep-sea sharks possess specialized organs known as the ampullae of Lorenzini, small pores distributed across the head and connected to internal receptors by channels filled with conductive gel. These structures are sensitive to minute electrical signals generated by the muscle activity of other animals. Each contraction produces a faint field that extends into the surrounding water, carrying information about presence and movement. The range of this sense is limited, but within that range, it is remarkably precise. A buried organism, concealed beneath layers of sediment, still produces electrical activity that can be detected through the substrate. The shark does not need to see or even disturb the surface to locate it. The signal passes through the water and the sediment alike, registering as a pattern that leads directly to its source. Chemical perception extends this reach over far greater distances. The deep sea contains gradients of dissolved compounds that spread slowly but persist over time, forming trails that can be followed across vast areas. Many organisms navigate primarily through these gradients, detecting slight differences in concentration and adjusting their direction accordingly. The water carries these signals outward, dispersing them but not erasing them, allowing a continuous path to form between source and receiver. Observations of the sleeper shark, Somniosus pacificus, reveal a capacity to follow such trails across tens of kilometers of open seafloor. In regions where physical features are minimal and visual cues are absent, it moves along these invisible pathways, tracing the distribution of molecules released from distant food sources. The path is not marked by any visible change in the environment, yet it remains consistent enough to guide movement over extended distances. Within the midwater column, another phenomenon becomes apparent through acoustic observation. A dense layer of organisms gathers at certain depths, forming a concentration so extensive that it reflects sonar signals as if it were a solid boundary. Early measurements suggested the presence of a shifting seafloor, rising and falling in response to unknown forces. Over time, it became clear that this apparent structure was composed entirely of living bodies, moving collectively through the water. This aggregation, known as the deep scattering layer, undergoes a daily vertical migration. As conditions change, the entire mass ascends and descends in unison, creating a pattern that spans large portions of the ocean. The movement is coordinated, not through vision, but through the combined influence of pressure, light intensity, and the presence of other organisms, each individual responding to local cues that produce a global pattern. Across these systems, perception extends beyond the limits of sight, forming a network of senses that interpret motion, electricity, and chemistry as primary sources of information. Each signal moves through the water in its own way, overlapping with others to create a layered environment that can be read without light. Time stretches differently where food does not arrive on any predictable schedule, where long intervals pass without contact, and survival depends on readiness rather than routine. In this environment, feeding is not a frequent act, but a rare event, one that must be resolved completely in the moment it appears. Bodies are shaped around this uncertainty, designed not for efficiency in repetition, but for success in a single encounter that may not come again for weeks. The black swallower, Chiasmodon niger, carries this principle into its structure. Its body appears unremarkable until the moment it engages with prey, when its jaws expand and its stomach begins to unfold. The connective tissues allow for extreme distension, enabling it to engulf organisms more than twice its own length and many times its mass. The boundaries of its form are not fixed but elastic, capable of reshaping to accommodate whatever it encounters. There is a record of one such encounter that extended beyond the limits of what the fish could sustain. A specimen was recovered at the surface with a snake mackerel contained within its body, the prey so large that digestion could not proceed at the required pace. As decomposition began, gases accumulated inside the enclosed space, inflating the swallower from within. Unable to release the pressure in time, it rose upward, carried by the expanding volume, until it reached a region where the internal structure could no longer hold. The event was not a failure of design, but the consequence of a system built to accept any available opportunity without restraint. A different configuration appears in the gulper eel, Uropharynx pelicanoides, whose mouth forms a large portion of its total length. When opened, it creates a broad cavity that extends outward like a pouch, enclosing water and anything suspended within it. This expansion is supported by a similarly flexible stomach, allowing the animal to process large volumes at once, rather than targeting individual prey items. The intake is not precise but expansive, capturing whatever is present in a single motion. At the opposite end of its body, a small luminous structure emits a faint red glow, positioned behind the main feeding apparatus. This placement suggests a strategy that draws attention away from the mouth, guiding movement toward a region where capture can occur without warning. The arrangement divides the body into two functional zones, one that signals and one that receives, connected through a shared extension of tissue and motion. The viperfish presents another variation, refining the mechanics of capture through structural modification. Its skull does not open in the usual downward motion, but hinges upward, increasing the angle of its gape far beyond what is typical in shallow water species. This movement allows the jaws to encompass a larger volume relative to body size, while the reinforced vertebra distributes the force generated during impact. The strike is rapid and direct, transferring energy through a structure designed to absorb and redirect it without damage. In the fangtooth fish, Anaplogaster cornuta, the emphasis shifts toward retention rather than expansion. Its teeth are proportionally among the largest in the ocean, extending beyond the limits of its jaw when closed. To accommodate them, the skull has developed sockets that receive the tips of the teeth, allowing the mouth to seal despite their length. The configuration ensures that once prey is taken in, escape becomes unlikely. The fish relies less on visual tracking and more on the detection of movement through pressure, locating targets through disturbances that propagate through the surrounding water. Across these forms, feeding structures reflect the same underlying condition, a scarcity that does not permit hesitation. Each adaptation increases the likelihood that an encounter, once initiated, results in a complete transfer of energy. The body becomes a mechanism for converting rare opportunities into sustained existence with little margin for error. These jaws, varied in shape and function, are built around a single constraint, the need to secure food when it appears, regardless of size or form. Beneath that shared requirement lies another layer, one that extends beyond individual encounters and into the broader system that supplies these rare moments of nourishment. Energy arrives here in fragments, not as a steady flow but as a slow descent of matter that began far above, where light still reaches and life gathers its strength from it. What supports the deep is not created within it, but delivered over time, carried downward through layers that gradually remove most of what was once available. The connection to the surface remains unbroken, though it becomes faint, stretched across distance and delay. Particles drift continuously from above, forming what has been described as marine snow. It is composed of the remains of organisms that lived in brighter waters, microscopic shells, fragments of plankton, waste products, and threads of organic material released into the surrounding currents. Each piece is small, often barely visible, yet together they create a constant, downward movement that links the upper ocean to the depths below. This descent is slow, measured at roughly 100 meters per day, allowing each particle to pass through multiple layers of consumption before reaching the seafloor. As it falls, it is intercepted repeatedly, grazed upon by organisms that extract what they can and leave behind what remains. With each exchange, the available energy is reduced, passed along in diminishing amounts through successive levels of the food chain. By the time these particles arrive at the abyssal plane, what remains is a fraction of the original energy. The principle governing this reduction is consistent. Only a small portion transfers from one level to the next, leaving the deeper layers dependent on what has already been consumed and processed multiple times. The result is an environment where energy is scarce, distributed thinly across a vast area, and where biological processes adjust to operate within these limits. Organisms living under these conditions tend toward reduced metabolic rates, conserving energy wherever possible. Movement is deliberate. Growth is extended over long periods, and lifespans lengthen in response to the slow pace of resource availability. The absence of abundant input shapes not only behavior but the structure of entire communities, aligning them with a system that cannot sustain rapid cycles of consumption and regeneration. Within this gradual system, an interruption can occur that alters the balance entirely. When a whale dies and begins to sink, its body carries with it a concentration of organic material far greater than anything delivered through marine snow. The descent ends at the seafloor, where the arrival introduces a sudden abundance into an environment defined by scarcity. The initial response comes from mobile scavengers that detect the presence of this mass through chemical signals spreading through the water. Species such as sleeper sharks and hagfish converge on the site, removing soft tissue at rates that can reach 40 to 60 kilograms per hour. Over the course of months, the majority of the flesh is consumed, leaving behind the skeletal structure as the primary remaining resource. This transition marks the beginning of a second phase, as organisms adapted to lower energy densities move in. Worms, crustaceans, and mollusks settle on and within the bones, extracting nutrients from the residual organic matter embedded in the structure. This stage extends over years, sustaining a community that remains localized around the remains of the original organism. As this material is further depleted, a third process takes over, driven by bacteria capable of accessing the lipids contained within the bones. Through their metabolic activity, these bacteria generate hydrogen sulfide, creating conditions similar to those found in other chemically driven ecosystems. This transformation supports a distinct group of organisms that rely on these compounds rather than on organic particles descending from above. The duration of this final stage can extend from 50 to 100 years, maintaining a localized system that continues long after the original structure has been reduced. Within this environment, species have evolved that are found nowhere else, adapted specifically to exploit the conditions created by this rare and concentrated source of energy. Such events stand apart from the general pattern, forming isolated zones where abundance briefly replaces scarcity. Around them, the broader seafloor remains governed by the slow descent of material from above, a steady but limited supply that shapes the pace and form of life throughout the deep ocean. The descent began without expectation of life, only the slow approach toward a seafloor thought to be defined by rock and silence. The submersible Alvin moved downward near the Galapagos Rift, carrying observers trained to read geology, not biology. Their attention was fixed on formations, fractures, and the shape of the crust, on structures that recorded the movement of the planet itself. The cameras were meant to document stone. What entered the frame instead did not match that intention. Rising from the seafloor were clusters of tall, pale forms, arranged densely around openings where water surged upward from beneath the crust. These structures were not mineral columns, but living organisms, tube worms extending up to 2.4 meters in length, their white bodies capped with deep red plumes. They stood in fields so crowded that the underlying rock was almost completely hidden, gathered around fissures where superheated fluid met the surrounding cold. Before this moment, the understanding of life's foundation was considered settled. Every ecosystem, without exception, was believed to depend on sunlight, directly or indirectly, through the process of photosynthesis. Plants and microorganisms captured light at the surface, converting it into chemical energy that supported every level below. This was not treated as a theory, but as a boundary condition for life itself. What appeared here did not fit within that boundary. The energy driving this system did not come from above, but from within the earth. Water, heated and enriched with dissolved chemicals as it passed through the crust, emerged through vents carrying hydrogen sulfide and other compounds. In the surrounding fluid, bacteria used these chemicals as a source of energy, oxidizing them to fix carbon into organic matter. The role usually filled by sunlight was replaced by a reaction rooted in geology. The tube worms at the center of this community embodied this shift. Riftia pachyptila possesses no mouth, no digestive tract, no opening through which food might pass. Instead, within its body lies an organ known as the trophosome, occupying a substantial portion of its internal volume. This structure houses dense populations of chemosynthetic bacteria sustained by the compounds delivered through the worm's tissues. The worm provides access to the necessary chemicals and oxygen, while the bacteria produce the organic molecules that support the host. In this arrangement, feeding is replaced by internal cultivation. The worm does not consume external material, but maintains a system that generates energy within its own body. The red plume, extending from its upper end, functions in part as a means of exchange, absorbing compounds from the surrounding water and delivering them inward to sustain the symbiotic process. Growth and survival depend on maintaining this balance, a continuous interaction between host and microbial population. Around these organisms, a dense assemblage forms, including mussels, crabs, and shrimp adapted to the same chemical environment. They cluster tightly around the vent openings, where the concentration of dissolved compounds is highest, and the temperature gradients are steep. The distribution is uneven, defined by proximity to the source, creating a pattern in which life is concentrated in narrow zones, while the surrounding seafloor remains comparatively sparse. Among these inhabitants, the shrimp Rimicaris exoculata exhibits a sensory adaptation aligned with the conditions of the vent. It lacks conventional eyes, yet along its back is a broad, light, sensitive surface capable of detecting faint emissions. The glow produced by the heated fluid may extend into wavelengths outside the visible range, and this structure appears suited to register that signal, allowing the shrimp to maintain position within a narrow band where conditions remain suitable. These communities do not persist indefinitely. Hydrothermal vents are transient features, their activity linked to geological processes that shift over time. When a vent becomes inactive, the supply of chemical energy ceases, and the organisms that depended on it must disperse, moving through the surrounding water to locate new sources that may lie far beyond immediate reach. Colonization begins again wherever conditions allow, establishing new clusters that repeat the pattern. Within this cycle, the vent forms a concentrated system, a place where energy emerges suddenly and supports dense populations for as long as it flows. Its existence reveals a mode of life independent of sunlight, grounded instead in the chemistry of the earth itself, sustained by reactions that occur beneath the seafloor and rise into the water above. There are places on the seafloor where nothing erupts, where no column rises and no visible disturbance announces what is happening beneath. The sediment appears undisturbed, the water still, and yet a slow movement is taking place, a steady release of chemical compounds seeping upward through the ground itself. These regions are known as cold seeps, though the name suggests an absence that is not entirely accurate. The water is not warm in the way vent fluid is, but the activity here is no less significant, unfolding at a pace that stretches across centuries rather than moments. From within the sediment, fluids rich in methane and hydrogen sulfide migrate upward, diffusing into the surrounding water without forming plumes or visible jets. There is no dramatic boundary between source and environment, only a gradual mixing that spreads outward through the seafloor. In this quiet exchange, bacteria find the same opportunity present in more energetic systems, using these compounds to generate organic material through chemosynthesis. The process is continuous, stable, and sustained over long periods, allowing entire communities to establish themselves around these subtle sources. Unlike the brief lifespans of more active systems, cold seeps can remain active for thousands of years, supporting generations of organisms that occupy the same location without interruption. The stability of the chemical supply creates a foundation for growth that does not require constant relocation. Mussels, clams, and other invertebrates cluster in these regions, their tissues housing bacteria that process the compounds rising from below. The relationship between organism and environment becomes steady and enduring, shaped by a flow that changes slowly over time. Within the sediment and surrounding layers, methane exists not only as a dissolved gas, but also in a more structured form. Under the conditions of high pressure and low temperature found at depth, methane molecules become trapped within crystalline frameworks of water ice, forming methane hydrates. These structures resemble solid ice, but contain within them large quantities of gas, locked in place by the surrounding lattice. They extend across wide areas of the sea floor, representing one of the largest reservoirs of carbon on the planet. The presence of these hydrates introduces another dimension to the system, storing energy in a form that remains stable under specific conditions, but can be released if those conditions change. The seafloor becomes not just a site of ongoing chemical exchange, but also a repository of material accumulated over long geological timescales, held in place by the pressure and temperature of the surrounding environment. Elsewhere on the seafloor, another phenomenon appears that alters the physical structure of the water itself. Brine pools form where dissolved salts concentrate to such a degree that the resulting fluid becomes denser than the seawater above it. This difference in density creates a boundary that behaves like a surface, allowing one body of liquid to rest upon another without immediate mixing. The result is a distinct pool, complete with a visible edge where the two layers meet. Crossing this boundary has immediate consequences for most organisms. The high salinity disrupts the balance of fluids within their bodies, drawing water out of cells and interfering with essential biological processes. Entry into the brine results in rapid failure of these systems, creating a stark contrast between the surrounding water and the pool itself. The separation is abrupt, defined by a transition that can occur over a distance of only a few centimeters. Along the edges of these pools, however, conditions allow for the formation of dense communities. Mussels and other organisms gather at the boundary, where chemical gradients provide the resources needed for chemosynthetic bacteria living within their tissues. The contrast between the two regions creates a narrow zone where life can persist in high density, supported by the same underlying processes that operate in other chemically driven environments. In one such location, discovered in the Gulf of Mexico, the accumulation of organisms that entered the pool and did not return has created a visible record along the shoreline. Crabs and other animals lie preserved at the edge, their bodies marking the boundary between two states of water that differ in composition but occupy the same space. The feature has been referred to as the jacuzzi of despair, a name that reflects the outcome observed at its margins rather than the processes that sustain it. Across these settings, the seafloor reveals structures that are not immediately apparent from a distance, systems that operate quietly yet support concentrated forms of life. The absence of dramatic motion does not indicate absence of activity, and the environments formed by these slow processes extend the range of conditions under which organisms can persist. At the edges of the great ocean basins, the seafloor does not remain level, but bends downward into narrow, elongated depressions that cut deep into the crust. These trenches mark the beginning of the hadal zone, a region that starts at 6,000 meters and extends to the deepest point yet measured, 10,935 meters, within the Challenger Deep of the Mariana Trench. The descent into these depths is not gradual in the way open water is, but confined, steep, and enclosed, creating an environment distinct from everything above it. Conditions here reach their most extreme expression. Pressure rises to 1,086 atmospheres, compressing every surface with a force that reshapes matter at the molecular level. Temperatures remain just above freezing, stable and unchanging. Light does not penetrate, leaving the entire region in complete darkness. These parameters combine to form a setting that appears incompatible with complex life, yet observations have shown otherwise. When a descent was made in 2012 using a solo submersible, the camera revealed organisms present on the sediment. Sea cucumbers moved slowly across the surface, shrimp navigated the water just above it, and microscopic life persisted within the layers below. This was not an isolated record. Decades earlier, in 1960, a bathyscaphe carrying Jacques Piccard and Don Walsh reached similar depths and reported the presence of a flatfish resting on the seafloor, positioned as if it were in an environment no different from any other. Within these trenches, a pattern emerges that diverges from expectations formed in less extreme settings. Amphipods, small crustaceans that measure only a few centimeters in shallow water, reach lengths of up to 34 centimeters in the hadal zone. Their bodies extend beyond what might be predicted under such pressure, growing larger rather than diminishing. This increase in size occurs consistently across species occupying these depths, suggesting that the conditions present here favor expansion rather than reduction. Several factors appear to contribute to this outcome. Lower temperatures, slow metabolic processes, extending the time available for growth over the lifespan of an organism. Oxygen levels in the deep water can remain relatively high, supporting sustained biological activity despite the surrounding constraints. The structure of the trench itself concentrates organic material, guiding particles downward and trapping them within the basin, providing a more consistent supply of resources than the surrounding seafloor. Among the organisms inhabiting this region, the snailfish appears again, not as a passive inhabitant, but as an active predator. At depths exceeding 8,000 meters, it moves through the water with controlled motion, locating amphipods and capturing them with apparent efficiency. Its body retains the characteristics that allow it to withstand pressure, yet its behavior reflects a role within the local ecosystem that extends beyond mere survival. The sediment covering the floor of these trenches forms a layer that can reach several meters in thickness, built up over long periods as particles accumulate from above. Within this material, traces of human activity have been detected, including industrial compounds that have traveled from the surface and settled into these remote regions. The same processes that concentrate organic matter also collect these substances, embedding them within the environment and introducing elements that were not present in earlier conditions. The presence of such compounds within organisms at these depths indicates that the isolation of the hadal zone is not complete. Materials introduced at the surface can move through the ocean and become incorporated into systems far below, carried along pathways that connect distant parts of the planet. The trenches, while physically separated, remain linked to the broader ocean through these continuous exchanges. Across this environment, life persists in forms that reflect both the constraints and the opportunities presented by the trench. Growth patterns shift, behaviors adapt, and the distribution of resources shapes the structure of communities in ways that differ from the open seafloor. The conditions that define the hadal zone do not eliminate life, but redirect its development, producing outcomes that challenge expectations formed elsewhere. The deep ocean was not always the place it is now, and its current character emerged through a shift that unfolded over millions of years. Roughly 34 million years ago, a change in the configuration of continents opened a passage between Antarctica and South America, allowing currents to circulate freely around the southern polar region. This movement altered global climate, initiating the formation of large ice sheets and gradually lowering the temperature of the water that filled the deepest parts of the ocean. Before this transition, the deep sea held warmth that no longer exists there, and its chemistry followed patterns shaped by that earlier state. The layers of water were less sharply divided, and the distribution of heat and nutrients differed from what is observed today. The conditions that now define the deep ocean, cold, stable, and isolated from surface influence, are relatively recent in geological terms, emerging after this reorganization of planetary circulation. As the environment changed, organisms that once occupied shallower regions encountered new conditions as they moved downward, whether through gradual expansion of range or shifts in habitat over time. These movements did not occur once, but repeatedly, across many lineages, each entering an environment that presented challenges unlike anything their ancestors had experienced. The descent into depth required adjustments that could not be borrowed from existing traits, but had to be developed through incremental changes across generations. Phylogenetic studies reveal that this process happened independently in numerous groups, with species from different branches of the evolutionary tree arriving at similar destinations through separate paths. The transition from shallow water to the deep required solutions to the same fundamental constraints: the increasing pressure, the lowering temperature, and the absence of light. Each lineage approached these constraints with its own variations, yet the outcomes often converged toward comparable forms. The snailfish family provides one example of this trajectory. While some members inhabit relatively shallow environments, others have extended their range into the deepest regions known to support vertebrate life. This expansion appears to have taken place within the last 20 million years, a relatively short period in evolutionary terms, indicating that the capacity to adapt to such conditions can arise more than once within a lineage given sufficient time and selection. Across unrelated species, similar features emerge not because of shared ancestry, but because of shared necessity. Bioluminescence appears in multiple groups through different chemical mechanisms. Eyes diminish or disappear where they no longer provide useful information. Bodies become softer, allowing pressure to distribute evenly rather than resist it. Pigmentation fades where it serves no function, and internal chemistry shifts to maintain stability under compression. These parallels reflect a process known as convergent evolution, where distinct lineages arrive at similar solutions when faced with identical problems. At the same time, traits that no longer contribute to survival begin to fade. Maintaining structures that require energy without providing benefit becomes disadvantageous in an environment where resources are limited. Over successive generations, such traits are reduced or lost entirely. Vision, coloration, rigid skeletal structures, and rapid metabolic rates all diminish where they are not required, replaced by features that align more closely with the conditions of the deep. This process can give the impression that organisms have become simpler, yet the changes reflect a refinement rather than a reduction. The removal of unnecessary components allows the remaining systems to operate with greater efficiency, conserving energy and maintaining function under constraints that leave little margin for excess. Each adjustment is shaped by the balance between cost and benefit, producing forms that are closely matched to their surroundings. Through this gradual transformation, the deep sea has assembled its communities not from a single origin, but from many, each contributing variations on a common theme. The resulting diversity reflects both the shared pressures of the environment and the different paths taken to meet them, forming a mosaic of adaptations that continues to evolve as conditions persist. Among bodies shaped by scarcity and restraint, a different pattern persists, one that does not simplify but reorganizes complexity into unfamiliar forms. Cephalopods occupy this space with a structure that distributes intelligence rather than concentrating it, allowing perception and response to unfold across the entire body. Their presence introduces a layer of behavior that does not rely solely on reflex or simple reaction, but on systems capable of processing information in parallel, each part contributing to the whole. The deep-sea octopus, Granodon boreopacifica, expresses this capacity through a life history that unfolds over an extended duration. A single female attaches her eggs to a stable surface and remains with them for 4.5 years, maintaining contact without interruption. During this period, she does not feed. Her movements are limited to small adjustments, circulating water across the eggs to supply oxygen and prevent accumulation of waste. The process continues through seasonal cycles that have no direct expression at depth, sustained entirely by internal reserves and the gradual expenditure of stored energy. As development progresses, the mother's body diminishes, her tissues providing the metabolic support required to maintain the brood. When the eggs hatch, releasing fully formed young into the surrounding water, the adult has reached the end of her capacity to sustain herself. The sequence concludes with her death, completing a cycle in which all available resources have been directed toward a single reproductive event. Underlying this behavior is a nervous system that differs from that of vertebrates in both structure and distribution. An octopus contains approximately 500 million neurons, yet only a portion of these are located within a central brain. The majority are embedded within the arms, forming localized networks that process sensory input and coordinate movement independently. Each arm is capable of responding to stimuli, exploring its surroundings, and executing actions without direct control from a central command. This arrangement becomes evident in experimental observations where an arm, separated from the body, continues to exhibit organized activity. It can react to touch, adjust its grip, and move in patterns consistent with feeding behavior, attempting to transfer material toward the position where the mouth would normally be located. The persistence of these responses reflects a system in which control is distributed rather than centralized, allowing function to continue even when connection to the main body is lost. Another lineage extends this pattern into a different mode of existence. The organism known as Vampiroteuthis infernalis occupies a position between squid and octopus, retaining characteristics of both, while forming its own distinct branch. Its structure has remained largely unchanged over long periods, preserving features that link it to earlier stages in cephalopod evolution. Its behavior diverges from the active predation typical of many related species. Instead of pursuing live prey, it gathers particles descending through the water column. Two elongated filaments extend outward, coated with mucus that captures organic material as it drifts past. These filaments retract, bringing the collected particles toward the mouth where they are consumed. This method aligns with the slow input of energy present in its environment, matching intake to the available supply without requiring pursuit or capture. When disturbed, the organism alters its form by drawing its webbed arms upward and over its body, exposing rows of spines that remain hidden during normal movement. At the same time, it releases a cloud of bioluminescent mucus into the surrounding water, creating a dispersed field of light that interferes with detection. The emission does not illuminate the organism itself, but introduces a pattern that disrupts the ability of others to track its position. Within its circulatory system, oxygen is transported by hemocyanin, a copper-based molecule that functions efficiently under the low temperatures and high pressures of its habitat. This adaptation supports metabolic processes that must operate within narrow limits, maintaining function where other systems would lose effectiveness. The giant squid, Architeuthis dux, represents another extension of cephalopod form, reaching lengths of up to 13 meters and occupying depths that remained largely unobserved for most of human history. Knowledge of its existence was derived from indirect evidence, remains recovered from the stomachs of predators, and fragments washed to the surface. For a long period, its living form remained unseen in its natural environment. In 2004, a camera captured a single image of a giant squid at depth, providing direct confirmation of its presence. The observation was brief, a momentary record within a vast and largely unobserved habitat. It did not resolve the full range of its behavior or distribution, but it established a point of reference within a system where direct observation is limited. The map fades quickly once it reaches depth, not because the terrain disappears, but because observation does. Estimates suggest that between 700,000 and 1 million species inhabit the deep ocean, yet fewer than 10% have been formally described. The remainder exist beyond classification, encountered only in fragments or not at all, their forms inferred from traces, brief encounters, or singular events that do not repeat. Each expedition into these regions returns with organisms that have no prior record, not as an occasional surprise, but as a consistent outcome. Sampling devices and remotely operated vehicles bring back specimens that do not match existing descriptions, adding to a catalog that remains incomplete despite decades of effort. While much of the surface world has been surveyed in detail, the deep ocean continues to yield new forms with each limited window of access. Among the most substantial of these forms is the colossal squid, Mesinicoteuthis hamiltoni, an organism that exceeds the giant squid in mass and carries structures adapted to its environment. Its tentacles bear rotating hooks rather than simple suckers, providing a means of securing prey in a medium where escape is possible in any direction. Its eyes reach diameters of up to 27 centimeters, the largest known among living animals, extending its capacity to detect faint signals in surrounding water. At that scale, the eye functions less as a general imaging system and more as a detector of specific events. The sensitivity allows it to register minimal light, including flashes produced by disturbances in the water caused by approaching predators. The movement of a large organism, such as a sperm whale, can generate bioluminescent responses in smaller creatures nearby, creating a field of light that signals motion from a distance. The squid's visual system appears tuned to detect these changes, providing information that may allow it to respond before direct contact occurs. Other species encountered in deep water reflect a continuity with much earlier periods. The frilled shark, Chlamydosalacus anguineus, retains a body plan that has changed little over tens of millions of years. Its elongated form and multiple rows of teeth, arranged in trident shapes, create a structure suited to capturing and retaining prey within a confined space. A live individual recorded off the coast of Japan in 2007 provided one of the few direct observations of its behavior, though the specimen did not survive long after being brought to the surface. The oarfish, Regalecus glesnae, occupies a different region of the water column but remains largely unobserved in its natural state. Reaching lengths of up to 8 meters, it moves through deeper layers with a body that extends in a continuous ribbon, its surface reflecting light in narrow bands. Encounters with this species often occur when individuals reach the surface in a weakened condition, providing rare glimpses of a form that otherwise remains out of view. Historical accounts from multiple cultures describe long, serpentine creatures appearing along shorelines, suggesting that these events have been recorded long before their biological origin was understood. Recent years have added further examples to this incomplete record. The identification of a small shark species in 2015, later referred to as the ninja lantern shark, expanded the known diversity within its group. Surveys conducted in the Mariana Trench in 2016 documented several new comb jelly species, each exhibiting structures not previously described. In 2021, physical evidence confirmed the existence of a beaked whale species that had not been recognized before, despite its size and presence within oceanic environments. These findings illustrate that discovery in the deep sea is not limited to small or obscure organisms. Large animals, capable of movement across wide areas, remain undetected until circumstances allow for observation. The limitations of current methods restrict the extent of exploration, leaving vast regions unexamined and many forms unrecorded. The deep ocean remains in a state where absence of evidence reflects absence of access rather than absence of life. Each new observation expands the known range of forms, yet the majority of the environment continues beyond direct study, its inhabitants existing within conditions that are only partially understood through the brief intervals in which they are encountered. For much of recorded science, the deep ocean existed more as an absence than a place. A region defined not by what was known, but by what was assumed impossible. Cold, pressure, and darkness were treated not as variables to be studied, but as limits that life could not cross. A boundary drawn from sparse observations and extended into certainty. Within this framework, the depths below a few hundred meters were imagined as a final, empty layer, a quiet conclusion to the living surface above. In 1843, Edward Forbes gave this assumption a formal structure, proposing what he called the azoic zone, a depth beyond roughly 550 meters where life was believed to cease entirely. His conclusion rested on dredging attempts that returned little from deeper waters, an absence interpreted as evidence rather than limitation. The idea settled into acceptance, not because it had been thoroughly tested, but because there was little means to test it further, and so it remained, repeated and reinforced for decades. The first clear fracture in this view came not from a planned biological survey, but from the maintenance of a telegraph cable. In 1860, a section of transatlantic line was raised from approximately 2,000 meters for repair, and when it emerged, it carried with it organisms attached along its length. Corals, worms, and mollusks had colonized the cable in complete defiance of the assumed boundary, their presence quiet but undeniable. The depth at which they were found did not align with the accepted limit, and so the limit itself began to dissolve. A more deliberate effort followed with the voyage of HMS Challenger between 1872 and 1876, a ship dedicated not to transport or communication, but to the systematic study of the ocean. Over the course of 130,000 kilometers, it conducted dredging operations across multiple depths, recovering thousands of specimens from regions previously unexamined. By the end of the expedition, approximately 4,700 new species had been cataloged, and the findings were compiled into extensive reports that continued to inform oceanographic research long after the voyage had ended. Decades later, the attempt to observe these environments directly took a different form. In 1934, William Beebe and Otis Barton descended in a bathysphere, a steel sphere with a diameter of 1.45 meters, lowered by cable into the water column. At a depth of 923 meters, Beebe described what he saw through a small window, relaying his observations by telephone to listeners at the surface. The images were not recorded visually, but through his words, capturing forms that had never been seen alive by human eyes. By 1960, the descent reached its deepest point when Jacques Picard and Don Walsh traveled to 10,916 meters in the bathyscaphe Trieste. The vessel carried them to the floor of the Mariana Trench, its viewing port only 7.6 centimeters thick, designed to withstand the immense pressure surrounding it. Communication with the surface relied on sonar, and the time spent at depth was brief, a short presence within an environment that remained otherwise undisturbed. The development of the submersible Alvin, operational since 1964, provided a more sustained means of exploration. Over more than 5,000 dives, it became a central tool in deep-sea research, contributing to discoveries that reshaped understanding of ocean life. It located hydrothermal vents in 1977, revealing ecosystems independent of sunlight, and later examined the wreck of the Titanic in 1986, extending its use beyond biology into broader exploration. Despite these advances, the scale of the ocean remains largely beyond reach. Crude submersibles can occupy only a small portion of the seafloor at any given time, and individual missions often survey areas measured in square meters within an environment that spans vast distances. Many discoveries arise not from targeted searches, but from encounters made while pursuing other objectives, fragments of a larger whole that remains mostly unseen. The effort to understand the deep sea continues within these constraints, each descent adding detail without completing the picture. The progression from assumption to observation has altered the understanding of what exists below, revealing that absence was never a property of the environment itself, but of the tools available to perceive it. A plane that stretches beyond any horizon lies far below the reach of currents that stir the surface, a level expanse where distance is measured not in landmarks, but in the slow accumulation of particles settling through water over spans of time that exceed memory. At depths between 4,000 and 6,000 meters, this terrain forms the abyssal plain, a continuous seafloor covering more than half of Earth's surface. A single ecosystem larger than any forest or desert, extending in quiet continuity across entire ocean basins. At first encounter, it appears nearly vacant. Movement is sparse, individual organisms separated by wide intervals of bare sediment, each one occupying its own small radius within an immense openness. A few tracks may cross the surface, faint disturbances marking where something passed hours or days earlier, then nothing again for long stretches. Yet the stillness is only apparent, because beneath that thin, visible layer, the sediment itself holds an activity that rarely reaches the eye. In some regions, this sediment has accumulated to depths approaching 800 meters, built from countless layers of marine snow that have settled, compacted, and transformed over hundreds of millions of years. Each particle carries a fragment of the surface world above, reduced and altered as it descends, until it becomes part of a slow, continuous archive embedded in the seafloor. Within these layers, microbial communities persist in densities that rival far more visible ecosystems, metabolizing what little energy remains and sustaining processes that continue long after the original material has lost its form. Scattered across this plain are fields of manganese nodules, rounded mineral formations that rest on the sediment like stones placed by an unseen hand. Each begins with a small nucleus, a fragment of bone or a tooth, and over time, layers of metal oxides precipitate around it. Their growth proceeds at a rate of 1 to 2 centimeters per million years, a pace so gradual that a single nodule embodies spans of time longer than entire geological eras familiar at the surface. When they occur together, they form extensive pavements, a pattern repeated across regions that no human has directly observed forming. Among these nodules and sediments, there exist organisms that stretch the definition of individuality. Xenophyophores, single-celled protists, can reach sizes of up to 20 centimeters in diameter, their bodies constructed from cytoplasm supported by frameworks of collected sediment particles. They have no tissues or organs, yet their internal complexity allows them to maintain structure and function across scales that resemble small, multicellular life. A single cell becomes visible, occupying space in a way that challenges expectations formed from smaller examples of cellular life. Across the surface, movement resumes in slow patterns. Sea cucumbers, known as Scotoplanes globosa, move in groups, sometimes numbering in the hundreds, advancing in the same direction across the sediment. Their bodies lift slightly as they progress, ingesting the substrate beneath them and extracting organic material and microbial life contained within. Their alignment reflects gradients that are not visible, chemical traces dispersed through the sediment, guiding collective motion without any central coordination. Within the remnants of larger events, another form of life emerges. Osedax worms inhabit the bones of fallen animals, extending root-like structures into the matrix and secreting acids that dissolve and access the lipids contained within. They possess no mouth or digestive tract, relying instead on symbiotic bacteria housed within their tissues to process the material they release. Evidence of their activity appears in fossilized bones dating back 100 million years, indicating that this method of life has persisted through epochs, adapting to hosts that changed over time while the strategy itself remained. The plane continues outward, its scale resisting full comprehension, its processes unfolding at speeds that rarely align with observation. Beneath the surface layer, within sediments that have recorded the passage of entire eras, microbial systems operate on chemical pathways that have endured since long before complex life took form, sustained by gradients and reactions that remain largely hidden from view. Beneath the soft sediment of the seafloor, below the reach of burrowing animals and the slow turnover of organic particles, there exists another layer of existence that does not reveal itself through movement or form. It is not visible as a community spread across a landscape, but as a presence embedded within the rock itself, occupying spaces so small and isolated that they seem, at first, incapable of sustaining anything at all. For a long time, this region was assumed to be sterile, a boundary where life yielded to geology. In 1987, that assumption was quietly overturned. Scientists drilling into subsea floor sediments expected to find a gradual decline in biological activity, ending in lifeless material as depth increased. Instead, at nearly 2 kilometers below the seafloor, they encountered living bacteria, cells that were not dormant remnants but metabolically active organisms, existing in rock that had been sealed away from the ocean for millions of years. The discovery revealed a hidden biosphere, not above or upon the Earth, but within it. This deep biosphere is now understood to be vast, extending through sediments and into the underlying crust, forming a reservoir of life whose total biomass may rival that of all organisms at the surface combined. The cells that inhabit this realm operate under conditions that differ fundamentally from those experienced in open water. Nutrients are scarce, energy is limited, and isolation is nearly absolute. Yet within these constraints, life persists, not in abundance of activity, but in persistence over time. The pace at which these organisms live is almost difficult to frame. Some populations divide only once every 1,000 years, their metabolic processes proceeding at rates that approach the limits of detection. They remain active, maintaining internal order and repairing cellular components, but the tempo of their existence aligns more closely with geological change than with biological rhythms familiar at the surface. Movement is replaced by endurance, growth by continuity. Their energy sources are drawn not from light or recent organic material, but from chemical reactions occurring within the rock. In some regions, water interacts with iron-bearing minerals in a process known as serpentinization, producing hydrogen as a byproduct. This hydrogen becomes a fuel, supporting microbial communities that rely entirely on this reaction, independent of any external input from the ocean above. In other cases, organisms metabolize ancient organic carbon buried within sediments, extracting the final traces of energy from molecules that have remained unchanged for tens of millions of years. Within this environment, there is no cycle of day and night, no seasonal variation, no direct connection to surface processes. Conditions remain stable over immense spans of time, allowing life to settle into a state of near equilibrium, sustained by slow chemical gradients rather than dynamic exchanges. The boundaries that define ecosystems elsewhere, temperature shifts, light availability, fluid movement, are absent here, replaced by a constancy that extends across epochs. The existence of this hidden biosphere has reshaped the understanding of where life can persist. It demonstrates that biological systems do not require sunlight, atmospheric exchange, or even proximity to the surface. Instead, they can arise and continue within the solid earth, drawing energy from interactions that occur naturally between water and rock. This realization has extended beyond the study of our own planet, influencing the way other worlds are considered, suggesting that life may exist in places previously dismissed as uninhabitable. As the exploration of the deep ocean has progressed, each layer has revealed systems that operate under different rules, expanding the boundaries of what is recognized as possible. The deep biosphere represents one of the most distant of these layers, not in terms of location, but in terms of conditions, a realm where life is defined not by visibility or movement, but by its capacity to persist, quietly and continuously, within the fabric of the earth itself. Long before any instrument touched the water, the deep ocean carried signals that moved through it with quiet efficiency, spreading outward in all directions, bending, reflecting, and returning in patterns shaped by temperature and pressure. Sound, more than light, defines this environment, traveling through seawater at roughly 4.5 times the speed it does through air, allowing distant events to remain connected across basins that would otherwise feel isolated. In the absence of visibility, vibration becomes a map, a medium through which space is measured and presence is known. At depths between 600 and 1,200 meters, the ocean forms a layer where the speed of sound reaches a minimum, shaped by the balance between decreasing temperature and increasing pressure. This region, known as the SOFAR channel, acts as a natural waveguide. Sound waves entering it are refracted back toward its center, held within the layer and carried across immense distances with minimal loss of energy. A signal released into this channel can travel thousands of kilometers, curving along the Earth's surface, linking distant points through a continuous acoustic pathway. In the 1940s, this property was identified and adapted for military use, allowing submarines to be detected across large distances. Yet the channel itself had been present long before it was measured, and its properties had already been integrated into the lives of animals that moved through it. Whales, navigating across entire ocean basins, produce low-frequency calls that enter this layer and propagate outward, maintaining contact with others far beyond visual range, their communication shaped by the same physical rules, that later instruments would describe. Occasionally, the ocean produces sounds that resist immediate explanation. In 1997, an array of underwater microphones recorded a signal in the southern Pacific that was unusually powerful, extending across a wide frequency range and exceeding the intensity of known biological sources. It became known as the bloop, a sound traced to a region near Antarctica, its origin uncertain and subject to investigation. For years, it remained unexplained, a recorded event without a clear source, until further analysis linked it to an icequake, a fracture within the Antarctic ice shelf releasing energy into the surrounding water. Other signals persist without resolution. Since 1989, hydrophones have detected the calls of an individual whale, producing sounds at 52 Hz, a frequency outside the typical range of known whale species. These calls are consistent in pattern and location, recorded year after year, yet no corresponding responses have been observed. The animal's identity remains uncertain, its presence known only through repeated recordings that continue without variation. Alongside these natural processes, a newer layer of sound has emerged, produced by human activity. Commercial shipping generates continuous low-frequency noise, while naval sonar systems emit powerful pulses designed to travel long distances. Seismic air guns, used in the search for underwater resources, release repeated bursts of sound into the water column, penetrating deep into the seabed. These signals overlap with the frequency ranges used by marine mammals, altering the acoustic environment in ways that extend far beyond their points of origin. Among the species affected are beaked whales, animals that spend much of their lives at depths exceeding 1,000 meters. During periods of intense sonar activity, groups of these whales have been observed stranding on shorelines, their tissues showing signs consistent with decompression injury. The pattern suggests a disruption in their normal diving behavior, a rapid ascent triggered by exposure to sound, altering internal pressures in ways their bodies are not adapted to withstand. The deep ocean continues to carry sound as it always has, its structure unchanged, its pathways intact. Yet, within that continuity, the character of what moves through it has shifted, layers of human-generated noise, joining signals that have traveled through water for millions of years, blending into an environment that remains largely beyond direct observation. Across spans of time that have outlasted continents and climates, the deep ocean has remained in motion without interruption, its systems adjusting slowly as the surface above shifted through eras of ice and heat. It endured impacts that reshaped life on Earth, absorbed changes in chemistry and circulation, and continued with a steadiness that seemed almost detached from events unfolding far above. In contrast, the presence that now reaches into these depths arrived only recently, measured not in epochs but in centuries, its influence expanding faster than the environment can respond. Along parts of the seafloor, wide nets are drawn across the sediment at speeds of 4 to 8 kilometers per hour, a method known as deep-sea trawling. These nets do not pass lightly over the surface. They cut into it, overturning layers that have settled undisturbed for long periods, and in doing so, they remove structures that require decades or centuries to form. Sponge fields and cold water coral assemblages, anchored in place and growing slowly in low-energy conditions, are broken apart in a single movement, leaving behind flattened ground where complexity once existed. The disturbance extends beyond what can be seen. A study conducted in 2021 estimated that bottom trawling resuspends more seafloor sediment each year than any other human activity, releasing stored carbon back into the water column. The quantity involved approaches the scale of emissions produced annually by the aviation industry, linking a process occurring far below the surface to broader changes in atmospheric composition. Material that had remained isolated within sediment layers becomes mobile again, entering cycles from which it had long been absent. In the Central Pacific, another form of extraction is being prepared. The Clarion-Clipperton Zone, an expanse between Hawaii and Mexico, roughly comparable in size to the continental United States, contains extensive fields of manganese nodules. These formations, developed over millions of years at rates of 1 to centimeters per million years, serve as both mineral resources and habitat. Their removal eliminates not only the nodules themselves, but the surfaces upon which organisms live, a loss that cannot be reversed within any time frame relevant to human planning. The process of collecting these nodules would also generate plumes of sediment that rise and spread through the water column, carried by currents across distances that extend far beyond the immediate site of extraction. As these particles settle, they cover surrounding areas, altering conditions for organisms adapted to specific substrates. The effect is not confined to a single location, but radiates outward, reshaping environments that were not directly targeted. At a broader scale, changes in ocean chemistry continue to unfold. As carbon dioxide dissolves into seawater, it alters the balance of carbonate ions, reducing the availability of the building blocks required by organisms that form calcium carbonate structures. Below the carbonate compensation depth, these structures begin to dissolve, affecting both the organisms themselves and the particles that contribute to the transfer of carbon into deeper layers. Simultaneously, shifts in temperature influence the distribution of oxygen within the ocean. Warmer surface waters hold less dissolved oxygen, and increased stratification limits the mixing that would otherwise transport oxygen downward. As a result, oxygen minimum zones are expanding at rates estimated between 3 and 8% per decade, gradually altering the conditions experienced by deep-living species. The organisms inhabiting these depths are adapted to stability, their life histories extending over long periods, their reproductive cycles unfolding slowly, their metabolic rates aligned with limited energy availability. The changes now occurring operate on timescales far shorter than those to which these systems are accustomed. Adjustments that once occurred over thousands or millions of years are now compressed into decades, introducing variations that cannot be matched by biological response. Within this contrast of timescales, the deep ocean continues to exist as it always has, its processes ongoing, its systems interconnected. Yet the additions introduced into it, physical disturbance, chemical alteration, and acoustic presence, move through an environment that has not previously encountered such rapid change, shaping conditions in ways that are only beginning to be observed and understood. Not as a distant frontier, but as a quiet contributor, the deep ocean has already entered human life in ways that rarely carry its name. Long before it was mapped or understood, its chemistry and biology began to shape tools that operate in laboratories, hospitals, and research centers, extending its influence far beyond the water that contains it. These contributions emerged not from a complete understanding, but from brief encounters, fragments brought back from environments still largely unseen. In hydrothermal systems, where temperatures exceed what most life can tolerate, bacteria such as Thermus aquaticus produce enzymes that remain stable under extreme heat. One of these enzymes, Taq polymerase, made possible the development of polymerase chain reaction, a method that allows specific segments of DNA to be copied rapidly and repeatedly. This process underlies modern genetic analysis, from disease diagnostics to forensic identification, enabling information encoded within cells to be read and amplified with precision. From entirely different organisms, other compounds have entered medicine. The venom of certain marine snails contains molecules that interact with the nervous system in highly specific ways. One such compound led to the development of ziconotide, a pain treatment used in cases where conventional options are no longer effective. Its mechanism does not rely on the pathways targeted by opioids, offering an alternative approach derived from interactions that evolved in the context of predation. Earlier still, compounds isolated from marine sponges contributed to the creation of cetarabine, among the first anti-cancer drugs developed from ocean-derived chemistry. Its application in treating certain leukemias reflects how molecular structures shaped in one context can be adapted to another, transferring from ecological function into clinical use. These transitions occur through careful study, each step revealing a connection between environments that at first seem entirely separate. The ability of some organisms to produce light has also been adapted into research tools. The luciferase system, responsible for bioluminescence, is now widely used as a reporter within living cells. When linked to specific genes, it allows researchers to observe when those genes are active, translating biochemical events into visible signals. This approach has been applied in studies ranging from cellular behavior to the progression of disease, enabling processes that would otherwise remain hidden to be tracked in real time. Structural solutions have also emerged from deep-sea organisms. Certain sponges construct their skeletons from silica, forming spicules that guide light with a level of efficiency comparable to engineered optical fibers. These structures develop under conditions of pressure and low temperature, demonstrating pathways for light transmission that do not rely on the materials or methods typically used in human manufacturing. In regions where hydrocarbons seep naturally from the seafloor, microbial communities have evolved the capacity to metabolize these compounds, breaking them down as part of their energy cycle. These organisms operate without external intervention, suggesting approaches to managing similar substances in other contexts. Their activity reflects processes that have been present for long periods, functioning independently of human presence. Each of these examples originates from a limited portion of the deep ocean, areas that have been sampled or observed through a relatively small number of expeditions. Beyond these regions lies a far greater expanse, one that remains largely unexplored. Within it, systems continue to operate, interactions unfold, and structures form, all without direct observation. The materials and processes already identified suggest that this larger domain contains a range of possibilities that has yet to be defined. The influence of the deep sea, then, extends quietly into systems far removed from its environment, not through deliberate design but through discovery and adaptation. It has contributed to methods, treatments, and technologies that shape modern life, even as its broader structure remains only partially known. Time does not pass here in the way it does above. It does not mark itself with seasons, or with cycles of light and darkness, or with sudden shifts in temperature that signal change. Instead, it settles into a near constant state where the pace of existence is governed by the slow arrival of energy and the steady persistence of cold. In this environment, life does not hurry. It extends. The Greenland shark moves through these waters with a rhythm that reflects that constancy. Its growth proceeds at roughly 1 centimeter per year, a rate so gradual that its body records decades as increments almost too small to perceive. It does not reach sexual maturity until around 150 years of age, and individuals have been estimated to live for at least 272 years. One specimen, examined through radiocarbon analysis, was dated to approximately 392 years old. That span places its early life in a world that existed centuries before the present, its movement continuing through events that unfolded far beyond the water it inhabits. The method used to determine this age relies on the eye lens, a structure that forms in layers, with its nucleus developing at birth. This core contains carbon that reflects the atmospheric conditions present at that moment, preserved without exchange over the animal's lifetime. By measuring the ratio of carbon 1:4 within this material, researchers can estimate the year in which the shark was born, linking a living organism directly to a point in time far removed from observation. Elsewhere, similar patterns of longevity appear in different forms. The orange roughy, a deep-sea fish that has been commercially harvested for decades, lives to around 149 years and does not begin reproducing until between 20 and 32 years of age. For many years, these characteristics were not fully understood, and fishing efforts removed individuals that had not yet contributed to the next generation. The result was a decline in populations that cannot be reversed quickly, because recovery depends on cycles that unfold over spans longer than most management systems are designed to accommodate. Across the deep ocean, this extended timescale shapes every aspect of life. Growth occurs slowly because energy is limited, reproduction is infrequent because encounters are rare, and lifespans lengthen because conditions remain stable. Without daily or seasonal variation, biological rhythms align with subtle changes in food availability and temperature, both of which shift only gradually. The absence of external markers does not eliminate time, but it transforms its expression. Within the bodies of these organisms, traces of time remain recorded. Otoliths, small structures within the inner ear of fish, accumulate layers as they grow, forming rings that correspond to years. In deep-sea species, these rings are often so compressed that they cannot be distinguished without magnification, each one representing a year of life condensed into a fraction of a millimeter. The record exists, but it requires careful examination to reveal it. This slow passage of time extends beyond individual organisms into the environment itself. Processes that would appear static on shorter timescales continue steadily, shaping conditions that persist across centuries. The accumulation of material, the transformation of chemical gradients, and the gradual adjustment of biological communities all proceed without interruption, guided by rates that do not align with immediate observation. In this setting, longevity is not an exception, but a consequence, emerging from conditions that favor endurance over speed. Life persists by extending its duration, by reducing its demands, and by aligning itself with an environment that changes so slowly it appears unchanged. The result is a continuity that stretches across spans of time that remain difficult to reconcile with more familiar measures. Beneath it all, the sediment continues to gather, layer upon layer, preserving within it a record that extends far beyond individual lifespans, a continuous archive of processes unfolding at a pace that has remained steady since long before complex organisms first appeared in the ocean. Long after motion fades from the water above, a quieter process continues below, grain settling onto grain in a descent too gradual to notice, yet persistent enough to build entire histories. The seafloor receives this steady fall without interruption, arranging each arrival into layers that rest upon what came before. Over time, these layers accumulate into a record that does not erode or scatter, but remains ordered, preserved in the stillness of depth. Each fragment that reaches the bottom carries with it a trace of the conditions in which it formed. Microscopic shells, composed of calcium carbonate, drift downward after the organisms that built them complete their brief lives near the surface. When they settle, they join countless others, forming a sequence that reflects the chemistry of the ocean and the atmosphere at the moment of their creation. The result is not a random accumulation, but a structured archive, with each layer corresponding to a specific interval in Earth's past. By extracting long cylinders of this sediment, scientists can examine the sequence directly, reading it as one would read the pages of a continuous document. Unlike records preserved on land, which are often broken by erosion, tectonic movement, or exposure to the elements, these deep layers remain largely undisturbed. Their continuity allows for a reconstruction of events across spans of time that extend far beyond human observation, revealing patterns that unfold over millions of years. Within this archive, foraminifera serve as precise indicators of past conditions. These single-celled organisms, present in the oceans for more than 500 million years, build shells that incorporate elements from the surrounding water. The ratio of oxygen isotopes within those shells shifts in response to temperature, creating a measurable signal that reflects the thermal state of the ocean at the time they lived. When these shells accumulate in sediment, they preserve that signal, allowing researchers to estimate ancient temperatures with remarkable accuracy, often within fractions of a degree. As these signals are traced through successive layers, broader patterns emerge. Variations in temperature, salinity, and atmospheric composition appear as gradual transitions or abrupt changes, marking periods of stability and disruption. Among these records is a distinct layer enriched in iridium, found at the same depth in cores collected from different ocean basins. This thin band corresponds to the impact that ended the Cretaceous period, a global event captured simultaneously across the planet, preserved in a uniform horizon that separates two profoundly different eras. Other layers reveal the opening and closing of ocean gateways, shifts in circulation patterns, and the advance and retreat of ice across continents. Each event leaves a signature in the sediment, encoded in chemical composition and particle structure. Volcanic eruptions deposit ash that settles into identifiable strata, while long-term changes in climate alter the types of organisms present, and therefore the composition of the material that reaches the seafloor. This record extends further back than most terrestrial archives, providing a continuous account that reaches into deep geological time. It forms a library that has been written slowly, without interruption, its pages composed of particles too small to see without magnification. The information it contains is not arranged for immediate interpretation, yet with careful analysis it reveals the sequence of events that shaped the planet. The accumulation continues in the present, each new layer adding to the record. The chemistry of the ocean, altered by processes now unfolding at the surface, is being incorporated into the shells that will eventually settle below. In time, these layers will become part of the same archive, indistinguishable in form from those that came before, yet carrying within them a distinct signature of this period. Beneath the quiet expanse, the sediment continues to gather, preserving each moment as it passes, extending a record that has endured through events of immense scale, maintaining its steady progression in darkness, while the surface above changes in ways that will one day be read from these layers. Far above the seafloor, where light still reaches and life gathers in thin abundance, an exchange unfolds continuously, invisible in motion, yet vast in consequence. Carbon moves from air into water, drawn into living cells at the surface, then released again in cycles that repeat without pause. What does not return immediately begins a slower journey, descending through layers of water that grow colder, darker, and more still, carrying with it the imprint of a changing atmosphere. At the surface, microscopic phytoplankton absorb carbon dioxide through photosynthesis, binding it into organic matter that forms the base of the marine food web. When these organisms die, or when they are consumed and repackaged into denser fragments, their remains begin to sink. This downward flux, known as the biological carbon pump, transfers carbon from the atmosphere into the deep ocean in a steady, continuous stream. It is not a sudden movement, but a gradual export, particle by particle, each fragment part of a larger process that operates across the entire ocean. As these particles descend, they are intercepted, broken down, and recycled by organisms at successive depths. Only a fraction continues downward, but that fraction is enough. Once carbon passes below 1,000 meters, it enters a realm where exchange with the atmosphere becomes extremely slow. There, it can remain isolated for centuries or longer, incorporated into deep water masses or eventually into the sediment itself, where it may be stored for millennia, before geological processes carry it further into the earth. This quiet transfer has been operating for hundreds of millions of years, moderating the concentration of carbon dioxide in the atmosphere and influencing global temperature. Without it, the balance between carbon sources and sinks would shift dramatically, and the atmosphere would contain far more carbon than it does today. The deep ocean, though removed from direct contact with sunlight, participates in climate regulation through this slow sequestration, acting as a long-term reservoir that stabilizes conditions at the surface. Alongside this biological process, the physical movement of water plays an equally important role. In polar regions, surface water cools, increases in density, and sinks, carrying with it dissolved gases, including carbon dioxide and oxygen. This sinking initiates a global circulation known as thermohaline flow, in which deep water spreads across ocean basins, traveling along the seafloor before gradually rising again elsewhere. The complete cycle unfolds over roughly 1,000 years, linking distant regions of the ocean into a single connected system. The water that occupies the deepest layers today began its descent long before modern industry, carrying with it a chemical signature that reflects the atmosphere of that earlier time. As it moves, it redistributes heat and dissolves substances, influencing conditions across the planet. This circulation is not rapid, yet its persistence allows it to shape climate over long timescales, smoothing variations and transporting energy in ways that are not immediately visible. As global temperatures rise, this circulation is beginning to change. Warmer surface waters reduce the formation of dense, sinking currents, weakening the downward transport that drives the system. With less efficient circulation, the ocean's capacity to absorb carbon and heat is altered, and the balance that has persisted for so long begins to shift. These changes unfold gradually, but their effects extend across regions and generations, influencing patterns that operate on scales far beyond immediate perception. The deep ocean is not separate from the climate system, but integral to it, participating through processes that connect surface activity with long-term storage and redistribution. Its role is not passive, but active, absorbing, transporting, and sequestering in ways that have maintained stability over geological time. Now those processes continue under new conditions, responding to changes that are occurring more rapidly than at any point in the recent past. The exchange persists, The descent continues and the circulation flows, but the balance they sustain is adjusting, slowly, in response to forces that extend from the surface into the depths, linking present activity with consequences that will unfold across centuries. The shift began not in the sky, but beneath a layer of water no sunlight could reach, where a discovery altered the assumptions carried for generations about where life could exist. For much of scientific history, the presence of a star was treated as a requirement, not simply a contributor, to any living system. Energy was expected to arrive from above, shaping climates, driving chemistry, and sustaining the processes that define biology. The surface of Earth provided the model, and that model extended outward into every early attempt to imagine life elsewhere. When ecosystems were found sustained entirely by chemical reactions within the planet itself, that framework loosened. Energy did not have to arrive from light. It could emerge from geological processes, from the interaction of water with rock, from gradients of temperature and chemistry that persisted independently of any external illumination. In these environments, organisms drew their energy from reactions involving hydrogen sulfide and other compounds, building entire communities that functioned without any connection to the surface. The realization was quiet, but its implications extended far beyond the ocean. If life could exist in such conditions here, then similar conditions elsewhere could no longer be dismissed. Attention turned outward, not to planets alone, but to worlds previously considered too distant from the sun to be viable. Among them was Europa, a moon of Jupiter encased in ice, beneath which lies a vast ocean kept liquid by the gravitational pull of its parent planet. The constant flexing of its interior generates heat, maintaining a subsurface environment where water remains in motion. Observations of its surface suggest that material from below is exchanged with the exterior, indicating active processes that may extend to the seafloor. Within that hidden ocean, the interaction of water with a rocky interior could produce conditions similar to those observed in Earth's deep environments. Chemical gradients, thermal energy, and circulating fluids would provide the ingredients necessary for systems that do not depend on light. The absence of a visible ecosystem does not imply absence of life, only that any such system would exist beyond direct observation in a region that has yet to be reached. A similar pattern appears at Enceladus, a smaller moon orbiting Saturn. Here, the presence of subsurface water is not inferred alone, but expressed directly through plumes that escape into space. These jets contain water vapor, organic compounds, and fine particles of silica, all consistent with interactions between liquid water and heated rock. Measurements have revealed molecular hydrogen within these plumes, a potential energy source for microorganisms, analogous to the processes that sustain certain ecosystems in Earth's oceans. The chemistry aligns closely with environments already known to support life, though separated by a distance that places them far beyond direct sampling. Even Titan, with its lakes of liquid hydrocarbons and dense atmosphere, expands the scope of consideration. Its chemistry differs from that of Earth, yet the existence of alternative energy pathways in deep environments suggests that life does not require identical conditions, only a stable source of energy and a medium through which chemistry can proceed. The range of possible habitats broadens, shaped not by the presence of sunlight, but by the availability of energy in any form. As these possibilities accumulate, the definition of a habitable environment shifts. It is no longer confined to surfaces warmed by stars, but includes interiors, oceans beneath ice, and regions where chemistry and geology intersect. The deep ocean provided the first clear example that life can organize itself around energy sources that do not involve light, establishing a model that can be applied beyond Earth. In this way, the understanding of life's requirements extends outward, guided by observations made in darkness. The patterns seen in one environment inform the interpretation of others, connecting distant worlds through shared principles rather than shared conditions. What was once considered exceptional becomes part of a broader framework in which life is understood as a response to energy and chemistry wherever they coincide. The deep ocean remains where these ideas were first grounded, its processes continuing as they always have, independent of observation. Yet its influence now reaches beyond its boundaries, shaping how the universe is considered and where attention turns when searching for systems that may exist unseen, sustained by forces that do not depend on light, but on the persistent interactions within matter itself. What appears distant and unfamiliar resolves, with enough patience, into something more recognizable, not because the forms become simpler, but because the patterns beneath them begin to align. The creatures moving through the deepest water seem shaped by forces that feel separate from the world above, yet the processes that sustain them are not separate at all. They follow the same chemical pathways, the same exchanges of energy, the same fundamental structures that govern every living system on Earth. All complex life traces its lineage back to the ocean. Long before anything walked on land or breathed air, the first coordinated movements, the first nervous systems, the first interactions between predator and prey unfolded in water. The deep ocean, though altered by time and pressure, remains connected to that origin. Its inhabitants are not deviations from the story of life, but continuations of it, shaped by different conditions, yet grounded in the same biological framework. Within each cell, the same membranes regulate the movement of ions, maintaining gradients that allow energy to be stored and released. The same molecules carry information, replicate structure, and drive metabolism. The process that produces energy in a deep-sea organism is not fundamentally different from the one that powers human muscle or maintains neural activity. Adenosine triphosphate, assembled and broken apart in cycles, links every organism through a shared biochemical language that has persisted across vast spans of time. The differences that emerge are not in the underlying system, but in how that system is expressed. Structures that appear unusual are often precise responses to specific constraints, refined over generations to operate efficiently under conditions that rarely occur at the surface. A jaw that extends beyond what seems possible, a body that yields rather than resists, a sensory system tuned to signals that do not involve light. Each reflects an adjustment to a particular environment. When viewed from a distance, these forms can seem unfamiliar, but when considered in terms of function, they reveal a consistent logic. The perception of strangeness arises from comparison, from placing one set of conditions against another and expecting continuity of form rather than continuity of purpose. Features that appear exaggerated or disproportionate are often exactly what is required to solve a problem defined by scarcity, pressure, or absence. Efficiency replaces excess, and structures are shaped not by aesthetic coherence, but by necessity. Across the deep ocean, this principle repeats, producing a range of organisms that differ widely in appearance, yet share a common foundation. The diversity observed so far represents only a small portion of what exists. Much of the environment remains unobserved, not in an abstract sense, but in a direct and practical one. Vast regions have never been seen, their inhabitants unrecorded, their interactions unstudied. The organisms that have been documented are those that intersected with exploration, not a complete representation of the systems that occupy the space. The boundary between what is known and what remains unobserved is not distant. It exists just below the layer where light fades, where visibility ends, and direct observation becomes more difficult. Beyond that threshold, the environment continues uninterrupted, extending across areas that have yet to be examined in detail. The absence of data does not indicate absence of life, only the limits of access. Within that unseen expanse, the same processes persist, shaping organisms according to conditions that differ from those at the surface, but follow the same principles. The deep ocean does not stand apart from the rest of life, but reflects it under a different set of constraints, revealing aspects that are less visible in more familiar environments. In this way, the deep sea functions as both extension and reflection, showing how a single biological framework can adapt to a wide range of conditions without altering its core mechanisms. The forms that result may appear distinct, yet they remain connected through the same underlying structure, part of a continuous system that spans environments from the surface to the deepest regions, unified by processes that operate regardless of light, temperature, or pressure. The record of where we have chosen to go is written not only in distances traveled, but in the places we have left unvisited. Maps of distant planets exist with a clarity that far exceeds our understanding of the terrain beneath our own oceans, a contrast that does not arise from neglect, but from the differing ways these environments reveal themselves. Solid ground, exposed to light and open to observation, allows its features to be traced from afar. Water does not. It absorbs, scatters, and conceals, turning even the most advanced forms of remote sensing into partial glimpses rather than complete views. Human presence beyond the atmosphere has been achieved through coordinated effort on a scale that reshaped technology and industry. By comparison, the journeys into the deepest parts of the ocean have been few, each one involving a small number of individuals descending into a region that remains largely unchanged by observation. The difference is not in significance, but in accessibility. Space offers visibility once reached. The ocean resists it at every level, requiring direct contact for even the simplest form of understanding. Signals that travel freely through air and vacuum diminish rapidly in water. Light fades, radio waves are absorbed, and radar becomes ineffective, leaving only narrow windows through which information can pass. As a result, the seafloor cannot be surveyed from orbit with the same resolution as a planetary surface. Each observation must be made in place, within the environment itself, by instruments or individuals capable of withstanding conditions that limit both time and movement. This creates a situation in which proximity does not translate into familiarity. The deep ocean exists on the same planet, within the same system of water that cycles through atmosphere and land, yet remains largely unseen. The boundaries that separate it from daily experience are not defined by distance, but by the properties of the medium itself. What lies beneath is not hidden by remoteness, but by the difficulty of observation, by the opacity that turns a continuous environment into one that appears fragmented. Within that concealed space, processes continue that have operated for immense spans of time. Chemical reactions sustain communities independent of light, structures form and dissolve, and layers accumulate that preserve the sequence of events across geological periods. These activities are not isolated from the rest of the planet, but integrated into its functioning, contributing to cycles that extend from surface to depth. The absence of direct observation has shaped perception, creating an impression that the unknown lies elsewhere, beyond the atmosphere, rather than within the boundaries of the Earth itself. Yet the largest continuous habitat on the planet remains largely unexplored, its extent exceeding that of any landscape encountered on land. The scale of what remains unseen is not theoretical, but measurable, defined by the proportion of the ocean that has yet to be directly examined. Human history has unfolded almost entirely at the surface, within a narrow range of conditions that support immediate survival. From that position, the ocean has been experienced primarily as an interface, a boundary between landmasses, rather than as an environment in its own right. Only recently has sustained attention been directed toward understanding what exists beneath that boundary, and even then, the progress has been incremental, constrained by the same factors that have limited observation from the beginning. This does not indicate a lack of curiosity, but reflects the paths through which curiosity has been expressed. Efforts have extended outward toward regions where visibility and measurement are more readily achieved, while the interior of the ocean has remained comparatively less accessible. The result is a distribution of knowledge that does not align with proximity, but with the ease of observation. Beneath the surface, the ocean continues uninterrupted, its processes independent of whether they are observed. The environment persists, shaped by forces that operate regardless of attention, maintaining systems that connect to the broader dynamics of the planet. The unknown within it is not an abstraction, but a present reality, occupying a space that is both near and largely unexamined. In this way, the direction of exploration reflects both possibility and limitation, revealing as much about the conditions that enable observation as about the subjects being observed. The ocean remains, extending downward from every point where water meets air, containing within it a continuity of processes that have yet to be fully described, existing alongside human activity, yet largely beyond direct experience, part of the same world, but not yet fully seen. Time in the deep ocean does not pass in the way it does at the surface, marked by seasons or cycles of light, but accumulates in layers and lineages that extend far beyond immediate perception. A single animal moving through those waters may carry within its body a history that stretches across centuries. Its slow growth, reflecting conditions that change so gradually, they are almost imperceptible within a human lifetime. The Greenland shark, drifting through cold northern depths, grows at a pace measured in millimeters each year, its existence unfolding across intervals that align more closely with historical epochs than with individual lives. Below it, the sediment forms a record that reaches further still. Each grain, each fragment of shell, represents a moment that has already passed, preserved in sequence. Some of those fragments belong to organisms that lived before the first flowering plants appeared, before grasses spread across continents, before birds took to the air. The accumulation continues, uninterrupted, creating a continuity that extends backward through intervals of time that exceed most other records available on Earth. The water surrounding such an animal has its own history, shaped by circulation patterns that move slowly across the globe. A parcel of deep water may have last been in contact with the atmosphere centuries ago, carrying with it the chemical signature of that earlier period. As it moves, it remains largely isolated, its properties changing only gradually, contributing to an environment that is stable over long durations. The deep ocean itself has maintained conditions similar to those observed today for tens of millions of years. During that time, global climates shifted, ice sheets formed and retreated, and entire groups of organisms evolved and disappeared. Marine mammals returned to the water from terrestrial ancestors, adapting once again to an environment that had remained present throughout their absence. Human societies emerged, developed technologies, and altered landscapes, all within a span that occupies only a small portion of the deep ocean's continuity. The lineages that inhabit these depths have persisted through multiple global disruptions. Mass extinction events that eliminated large proportions of marine species altered the composition of life, yet did not erase it entirely. Survivors carried forward traits that allowed them to endure, and over time, new forms emerged from those remnants. The current inhabitants of the deep ocean are part of that extended history, their existence shaped by conditions that have tested biological systems repeatedly. The scale at which these processes occur differs significantly from the timescales that structure human experience. Events that define human history, whether lasting decades or centuries, represent brief intervals when viewed against the backdrop of millions of years. The continuity of the deep ocean does not depend on rapid change, but on persistence, on processes that operate steadily over long durations. Within this context, the forms observed in deep-sea organisms reflect solutions developed under specific constraints. Features that may appear unusual when compared to surface organisms correspond to functional adaptations that address challenges associated with pressure, limited resources, and absence of light. These traits are not arbitrary, but the result of selective pressures acting over extended periods, refining structures to operate effectively within a given environment. The cumulative effect of these adaptations is a diversity of forms that, while varied in appearance, share a common basis in biological processes. Each structure, whether related to feeding, movement, or sensing, corresponds to a particular set of conditions, demonstrating how life can adjust to different circumstances while maintaining underlying continuity. The deep ocean, in this sense, serves as a record of responses, documenting how organisms have adjusted to environments that differ significantly from those at the surface. It reflects a range of solutions that have emerged over time, each aligned with specific challenges, each contributing to the broader understanding of how life persists. Through this extended perspective, the deep sea reveals a continuity that integrates biological, chemical, and geological processes, linking individual organisms to a history that extends far beyond immediate observation, forming a system in which present conditions are connected to past events through layers, movements, and adaptations that unfold across vast intervals of time. Far beneath the wide surface of the Pacific, where the last traces of light dissolved long ago, a small body drifts without hurry, suspended in a column of water that has known only darkness for millions of years. An anglerfish moves through that space, its lure suspended ahead of it, emitting a steady blue-white glow that never flickers, never fades, never rests. The light is not a choice but a condition, a partnership written into its biology, maintained by bacteria that convert chemistry into illumination. It has never seen a horizon, never encountered the shifting brightness of day, never known the cycle that defines life above. It exists entirely within a continuous night that does not begin or end, a darkness that stretches outward in every direction and persists beyond the boundaries of its brief life. Below, at depths where pressure increases beyond the limits of engineered structures, another form moves with equal calm. A snailfish glides just above the seafloor, its body soft, translucent, almost without resistance to the surrounding weight. The forces that would crush rigid materials are distributed through its tissues, absorbed and balanced, becoming simply part of the environment it inhabits. It does not resist the pressure, it lives within it. Shaped by it, its existence defined by conditions that remain constant across spans of time that far exceed individual experience. Deeper still, within the sediment that fills the narrow trenches, processes unfold at a pace that approaches stillness. Microorganisms embedded in the layers continue their activity, drawing energy from compounds that have remained buried since long before the present arrangement of continents was established. Their metabolism proceeds so slowly that change becomes difficult to perceive, yet it continues, steady and uninterrupted, linking present conditions to events that occurred millions of years in the past. The sediment itself becomes both environment and archive, holding within it the remnants of organisms that once moved through different oceans under different skies. The deep ocean operates without reference to surface cycles, without reliance on sunlight, without the variability that characterizes more familiar environments. Its continuity does not depend on observation or interaction, and its processes proceed whether they are understood or not. Long before any human presence, these systems were active, circulating energy, transforming matter, sustaining life forms adapted to conditions that remain largely inaccessible. The absence of light does not indicate absence of activity, but rather a different framework in which that activity occurs, governed by chemistry, pressure, and time. In that setting, the forms that emerge are not deviations but outcomes, each feature corresponding to a requirement imposed by the environment. Structures that appear unusual when viewed from the surface align precisely with the demands placed upon them, refined through countless generations. The anglerfish's light, the snailfish's softness, the microbial persistence within sediment, all represent solutions that have been tested across intervals far longer than recorded history. What becomes visible through this continuity is not fragility but endurance, not isolation but integration within a system that extends across the entire planet. Life is not confined to narrow conditions, but occupies a wide range of environments, adapting its mechanisms to operate under varying constraints. The deep ocean demonstrates this capacity in its most extended form, maintaining biological processes in places where external energy is limited and change is slow. The darkness itself remains constant, not as an absence but as a condition that shapes every interaction within it. It existed before the current inhabitants, and it will persist after them, continuing to frame the processes that occur within its reach. Each organism that passes through that space participates briefly in a sequence that extends far beyond individual lifespans, contributing to a continuity that does not depend on any single presence. At the surface, time is often marked by rapid change, by events that define shorter intervals. Below, time accumulates, layering events into a record that extends across geological scales. The difference between these perspectives does not alter the underlying processes, but it does shift the frame through which they are understood. The ocean remains, steady and unbroken in its depths, carrying forward the same processes that have sustained it for millions of years, a vast and continuous system moving quietly beneath the surface, present now as it has always been, and continuing onward without interruption